Ladies and gentlemen, on behalf of Director CSIR NST, it is with immense pleasure we welcome you all for the National Science Day celebrations at CSIR NST. National Science Day is celebrated annually in the country on February 28 to commemorate the discovery of Raman effect by Sir C.V. Raman in 1928, for which he was awarded Nobel Prize in 1930. The day aims to foster scientific temper, promote awareness of scientific achievements, and encourage innovations across the country. The theme for this year's National Science Day is Women in Science, Catalyzing Viksit Bharat, which aligns with the government's vision of Nari Shakti in STEM to boost national development. We are honored to have among us Professor Raghavan Varadarajan from IISc Bengaluru as the chief guest for the National Science Day celebration at our institute. May I now request our director, Dr. C. Anantharamakrishnan, to kindly accompany the chief guest, Professor Raghavan Varadarajan, to the dais. I also request Dr. T.P.D. Rajan and Dr. Joshi Joseph to accompany the dignitaries. Thank you, sir. I now invite Dr. T.P.D. Rajan, Chief Scientist and Head, Human Resource and Academic Division, to kindly deliver the welcome address.
Very good morning to all. Distinguished Chief Guest of the Day, Professor Raghavan Varadarajan, Professor Molecular Bioscience Unit, Indian Institute of Science, and President, Indian Academy of Sciences, respected Director, Dr. C. Anantharamakrishnan, Dr. Joshi Joseph, Dr. Jayamurthy, dear esteemed scientists, staff, dear colleagues, research scholars, students, distinguished guests and participants. It is my great privilege and honor to welcome you all to the celebration of National Science Day today here at CSIR NIST. I start with the famous quote of Sir C.V. Raman: "Ask the right questions and nature will open the doors of her secrets." National Science Day is observed every year to commemorate the discovery of Raman effect by Sir C.V. Raman, a milestone that placed Indian science prominently on the global map. This day reminds us not only our rich scientific heritage, but also our responsibility to shape the scientific future of our nation. The theme for this year is Women in Science, Catalyzing Viksit Bharat, is both timely and transformative. As India advances towards becoming a developed nation, a Viksit Bharat, the role of women in science, technology, innovation and leadership becomes central. Women scientists are not merely participants in progress, they are powerful catalysts driving discovery, policy, entrepreneurship, and societal transformation. We are very much honored to have with us as chief guest Professor Raghavan Varadarajan, an eminent scientist whose contribution to molecular physics and scientific leadership has significantly influenced Indian science. He is recognized for his contributions to understanding the relationship between protein sequence, structure and stability, and its application to design immunogens that elicit protective antibodies against HIV-1, influenza, and SARS-CoV-2. We look forward to the exciting National Day Science Lecture, which I am sure will inspire young minds and energize the researchers. I kindly request our director to welcome our chief guest with a bouquet of flowers. Thank you, sir. We are very much privileged to have our director, Dr. C. Anantharamakrishnan, who will deliver the presidential address today. Under his dynamic leadership, CSIR NIST continues to strengthen interdisciplinary science and innovation aligned with the national priorities. We had a great year under his leadership, celebrating the Golden Jubilee of CSIR NIST, laying a foundation stone by Honorable Prime Minister to CSIR NIST Innovation, Technology and Entrepreneurship Hub at Thonekal, transforming various technologies such as transforming pathogenic biomedical waste into value-added soil additives, very popular designer rice technology, instant coffee crema technology, plant-based leather additives, osmobites, aluminum magnesium scandium alloy for aircrafts, Jaivam, bio-based binders, and many more. We have great pleasure in welcoming you, sir, for this program. I extend my heartfelt welcome to Dr. Joshi Joseph, Jigyasa Coordinator, Dr. Jayamurthy, ACSR coordinator, who are the pillars behind organizing this event, along with the HRD members and other colleagues. I extend a warm and heartfelt welcome to all our beloved colleagues, staff, students, distinguished invitees, women scientists who are the torch bearers of today's theme, and also all the participants who have gathered here to celebrate the spirit of science. I also congratulate all the prize winners for the National Science Day celebration. Let this day inspire us to foster curiosity, champion inclusivity, and work collectively towards building a scientifically empowered and developed India. Thank you all, and I wish you all a meaningful and inspiring National Day celebration. I conclude the welcome address again with the quote of C.V. Raman. Every great discovery begins with an idea, nurtured by the passion to delve deeper into unknown. It is not enough to see beauty, one must also understand the scientific principle behind it. Thank you.
Thank you, sir. Dr. C. Anantharamakrishnan is a leader who inspires by example. Under his visionary leadership, CSIR NIST has sharpened its strategic focus on food technology, agro-processing, healthcare, biomanufacturing, environmental technologies, and sustainable innovations in material sciences. His efforts have not only propelled the Institute's excellence in research, but also expanded its societal impact by nurturing strong collaborations across academia, industry, and public health sectors. Dr. Anantharamakrishnan has been bestowed with first ever Rashtriya Vigyan Shri Puraskar in Agriculture Sciences by the Government of India, and also the Tata Transformation Prize instituted by Tata Foundation and New York Academy of Sciences. We now request our Director, Vigyan Shri Dr. Anantharamakrishnan, to graciously greet the gathering with this presidential address.
National Science Day greetings to everyone. Most respected Professor Raghavan Varadarajan, Dr. T.P.D. Rajan, Dr. Joshi Joseph, Dr. Jayamurthy, and all my colleagues, friends, students, and other than CSIR NIST students who are come for today, open day for the Science Day at CSIR NIST. Good morning to everyone. Indeed, a great honor to have a Professor Raghavan Varadarajan, one of the pioneer scientists in the country, President of Indian Academy of Sciences, Bangalore. Recently, 1 year back, both we got together Tata Transformation Prize, and the way in which science he pursued is entirely is a different spectrum. Today, you may be witnessed the way how he pursued the science. His dream to make vaccines more affordable. That is the idea what he floated for the Tata Transformation Prize. Maybe it takes some time, but the dream definitely one day will come true. Thank you so much, sir, accepting our invitation to being with us today. On behalf of CSIR NIST and my own behalf, I am welcoming you for this day. National Science Day, as mentioned, we have celebrated the discovery. That discovery, without Raman, no other lab throughout the globe, they can't run the science. That's the power of science. That's the power of Indian science. He pursued everything in India. He born in Trichy, whole career in India, and when he discovered with a very, very limited facility. Today, we want all the facility, money, everything, but still we are not able to achieve the what Dr. C.V. Raman has achieved it. That's what we wanted to cultivate, that type of scientific temperament to all our young minds. Today, we are talking about Indian science. Yes, we world number 3rd in the number of publications. 278,000 papers we published last year, next to China and US. So one of the highest number of rate of publications if you look at, better than other 2 nations. We are continuing to do that. Last year more than 90,000 patents filed, up by 28% higher. This is the direct impact. Indirect impact, if you look at last year, 350 million metric ton of the food grains we produced, out of which if you look at 150 million tons of the rice, 115 million tons of the wheat and maize, and 240 million tons of the milk. It's a huge amount of the food grains or the milk. We are world number 1 and 2 most of the commodities. What is the scientific intervention if you ask? Look at every year. We never expected 350 million is going to achieve now. 10 years back, we are very, very less. We thought of The productivity we can't increase, but we could able to increase the new varieties developed, including the oil seeds. If you look at the ISRO or defense, Chandrayaan or all the whatever the innovation has come in the ISRO, Aditya L1, now we wanted to Gaganyaan, PSLV, all these things, the without scientific temperament and contribution of the scientist, we would have not achieved this far. Even we wanted to make it non fossil fuel energy for our country 50% by 2050 30. That's what original target. Today we achieved 246 gigawatt of power from the non fossil fuel. If you go back and each and every where its source are coming, scientific intervention has come. Either wind energy or the solar energy, everywhere our footprint is there, indirect effort. 5 years before we achieved the 50% of that, because India is the third largest consumer of the energy and one of the rapid need of energy throughout the globe. All these things gives only one statement: without scientific power, no other nation or any nation can grow faster what we achieved today in India. India is the best example of combination of all together. UPI transaction, we developed the UPI system. Today recently AI Impact Summit demonstrated what India can do that. That also moving on the faster mode. In this scenario, basic science, fundamental science and the applied research, all combination of the AI, how is going to be in future. That's what we need to look into that. Hope the Science Day we are celebrating the success of past, also future, where we need to go there. And today we are very, very happy to listen from the one of the finest. I said I can't say that you listen his talk, you know that what is the science. That science always as the fundamentally we need to develop for the our human benefit. Either vaccines or the molecular biophysics, all are entirely different spectrum. Thank you, sir, for coming to NEST, and morning sir could able to visit some of the facilities. We want a collaboration with the IIC Bangalore, especially his lab. I think our bioscience department will use his presence at our institute, and also all the students, large number of students today is attending to listen his lecture. Also we are going for live today for exclusive live for this presentation. Thank you so much. Once again, National Science Day greetings to everyone.
Thank you, sir, for the inspiring address. Professor Raghavan Varadarajan is a renowned professor in Molecular Biophysics Unit at Indian Institute of Science, Bengaluru, and he is currently serving as the President of Indian Academy of Sciences as well. He earned his Masters from IIT Kanpur in 1982 and completed his PhD in Stanford University in 1988. And he has, followed by that, he has done postdoctoral at Yale University. He joined IASC in 1992, where his research has made significant contributions to protein stability, folding and designing, and including structure-guided vaccine and immunogen development for influenza, HIV and SARS-CoV-2. He also serves as a honorary professor at JNCSR and also chairman of the scientific advisory body Thermae Novo Biogenics. He is a recipient of several prestigious fellowships and awards, which includes B.M. Birla Science Award, Shanti Swarup Bhatnagar Prize, AIDS Innovation Grant Award by NIH, G.N. Ramchandran Gold Medal, J.C. Bose Fellowship, and prestigious Tata Transformation Prize. And he has played a key leadership roles in Indian science over the past decade. May I now respectfully invite Professor Raghavan Varadarajan for the National Science Day address. So please.
So, thank you, Dr. Ananda Ramakrishnan, for the very kind invitation to speak here, and I've really enjoyed the little bit that I've seen of NIST so far. This is my first visit here. So today I'll spend a few minutes telling you the next maybe half an hour or so, telling you about some of the work that has been ongoing in my lab over the past several years. So since this is a relatively heterogeneous audience, I'll say a few basic things about proteins and vaccines and vaccine design. And in case this is, at too low a level for some of you, please bear with me. Okay. So as you all know, proteins are biopolymers. They have 20 different kinds of amino acids that make up the protein chain, and typically they're on the order of about 100 to 500 amino acids long. And we can infer the sequence of the protein from the corresponding gene sequence, and because it's become so easy and rapid to sequence DNA, we now have a huge amount of information on the sequences of many different genes and proteins. Now most proteins have well-folded three-dimensional structures, and these are important for the function. There are parts of proteins or entire proteins which don't really obey this rule, but many of the proteins that we will be concerned with have well-defined three-dimensional structures. Now, it's possible nowadays to fairly accurately predict the three-dimensional structure of the protein from the amino acid sequence and therefore from the gene sequence, and this advance really came about because of the large amount of sequence data that was available in the public domain, as well as the protein structures that had been determined by crystallographers and subsequently NMR and cryo-EM over many decades, which were all publicly available. And then a few years ago, DeepMind took advantage of this and the advancements in AI and machine learning to develop AlphaFold. Now, in addition to the structure of the protein, another important property is its stability. So the stability of the protein is defined quantitatively in terms of the free energy difference between the folded and the unfolded states of the protein. So most proteins have largely two states, a folded compact state. Does this work here? Okay. Yeah. So, so just representing in a cartoon fashion the folded and the unfolded states, and for these, this free energy of unfolding is a property, is a function of the temperature of the protein, okay? And it's independent of the protein concentration. So if you plot the stability of a protein as a function of temperature, you get something called the protein stability curve. And many proteins are maximally stable around room temperature. Okay. So now I'll talk about immunogen design. So with the pandemic which we have all lived through a few years ago, you're all familiar with viruses and vaccines to some degree. The way that the immune system protects against viral infection initially is through neutralizing antibodies. And neutralizing antibodies are, I think you all know what antibodies are. They're molecules produced by the immune system that circulate in blood, and they are directed against the major viral surface proteins, blood and tissues. And many viruses are enclosed by membranes, right? Membranes are lipid, which are made up of lipid molecules, and lipid molecules are negatively charged, and we have lipids both on the surface of the cells and on the surface of many viruses. So now, in order for the infection to occur, the virus has to go inside the cell. And when the virus approaches the cell, there is a repulsion between the negatively charged membrane on the virus and the negatively charged membrane on the infecting cell. So in order to be able to infect the cell, this negative repulsion has to be overcome. and it's overcome in part because the viruses have proteins on their surface which are in what are called metastable shapes. So which means that they don't have a single shape, they have more than one shape, and the shape that is on the virus is actually not the most stable shape. During the process of infection, and I'll illustrate this by in a slide or two, they undergo a big change in their shape and go to the low energy conformation. And so going from this highly unfavorable shape to the favorable shape releases a lot of free energy, and this free energy is used to promote fusion of the viral and cellular membranes. So in the process of vaccine design, we need the viral surface protein to adopt the pre-fusion conformation. And besides neutralizing antibodies, there's another branch of the immune system called the cellular immune system, which also contributes to protection. So what happens is there are three broad branches of the immune system. The first is the innate immune system, which directly recognizes in a non-specific way foreign objects. The second is the humoral immunity, which is the antibodies that we talked about. And the antibodies, they bind to the infecting organism before it can infect a cell, and they prevent the infection. However, if the infectious agents gets past these two branches of the immune system, it's able to infect cells and replicate inside cells. Now, when the virus is replicating inside cells, it also has to come out. And so the viral proteins or fragments of the viral proteins become displayed on the surface of the cell, and then the cellular immunity recognizes these infected cells and targets them for destruction. So one of the viruses that we work on is something called respiratory syncytial virus, which causes over 100,000 deaths a year in infants and young children, and 97% of these deaths are in low and middle income countries, including India. So on the surface, there is this important protein called the F or fusion protein, and the fusion protein, as the name indicates, helps the infection by allowing the viral membrane and the cellular membranes to fuse. And in that process, it undergoes this big change in shape. Now, because this is the protein that's in the native form on the surface of the virus, then the antibodies that we want, which will block infection, should bind to this shape. Okay? This shape happens after the infection is too late to stop. Okay? So having antibodies against this shape is at best useless, and in the particular case of RSV, they actually enhance infection. I don't have time to explain the mechanisms, but when an RSV vaccine was initially made in the 1960s, it was made the way many vaccines were made at the time and still continue to be made. The virus was isolated and it was chemically inactivated, and that was used as the vaccine. And in clinical trials, when this inactivated virus was given to children, many of them fell sick, and a couple died. Okay, and they fell sicker than the children who were not given the vaccine or who were given a placebo. And people didn't understand why this was happening, and so for over 60 years, there was no RSV vaccine. So a vaccine became available in 2023 when some of these factors were better understood, and that's now available in the US and Europe and other developed countries, but the cost is about $300 a dose, which is unaffordable for people here. It's also only available for adults and the elderly, including pregnant women. So it's not available for children, because in children, when they tried one of the vaccine formulations, again the children became sick. So, you know, there's a lot that we still don't understand. Now, as an example, I'll talk about some of the work we did with during the COVID time. And although COVID is largely gone, but it's not completely gone, and people do continuously get infected, and the elderly and those with comorbidities, and there are a very large number of people now who have comorbidities, including diabetes, they really should be having vaccines, though they are not available in India. And all the current vaccines for this, as well as for many other diseases, they require refrigeration, and many require frozen storage. So as a result of this, the worldwide vaccine coverage during the pandemic was very non-uniform and remains to be uniform. So one of our efforts is to develop safe, efficacious and inexpensive vaccines for viruses. So COVID-19 was caused by SARS-CoV-2, which is an example of a sabecovirus, and there are many other sabecoviruses which can potentially cause pandemics. So we worked on these, we worked on flu, RSV, and this is what a large part of what we do. So the primary tool that we use and help to develop is something called deep mutational scanning. So it turns out nowadays it's very easy to make a mutant of a gene where you change a specific base in the gene, one or more bases. And you can do this in a parallelized way in high throughput. So if you have one gene, you can make thousands of variants of the gene in a relatively short period of time. And now you can clone this collection of mutant genes, and you can screen them for activity. And then you can get the sequences of the library. This is that mixture of mutated genes before and after you do the selection. And this, in a single experiment, allows you to tell the contribution of each base in the gene or each amino acid in the protein to the property that you're measuring. And this could be, for example, if you have an enzyme, it could be the activity of the enzyme. If you have a virus, it could be the fitness of the virus, its ability to infect cells, and so on and so forth. Okay. So this is the first, it's now called deep mutational scanning, and we did the first deep mutational scan of an entire protein in 2012. And then we mapped it onto the known structure of the protein, and you can see that the active site immediately stands out. And over the years, we've used it for many, many different purposes. But today, I'm going to focus on how we used it for protein stabilization. Okay. So now, I'll go through this a little quickly for lack of time, but the idea is very simple. It turns out that if you have a gene of interest and you want to study the function of the protein, you can display it on the surface of either bacteriophages, or in our case, we use yeast. Okay? So you can display the protein on the surface of a yeast cell. There's standard methodology for doing this, and then you can come in with a binding partner of the protein and attach something that is detected by a fluorescent antibody both to the binding partner and to the protein itself. And then you use a technique called fluorescence activated cell sorting, and this allows you to look at both the amount of expressed protein on the surface as well as how well it's binding, any target of interest. Now, when you make a mutation in the protein, as I said, proteins are well-folded, many of them well-folded globular structures. So when you make a mutation in the interior of the protein, very often it loses its stability, and if it's not stable, then it doesn't bind its binding partner because it gets degraded. So we can now introduce this kind of destabilizing mutation into the library that we have already made and look for mutations which suppress the unfavorable properties of this mutation. And we found out that when we have suppressors that are far away from the original mutation, these tend to be stabilizing mutations. So far, there aren't general ways of predicting which mutations we should make to have the protein more stable with very high accuracy. So despite all the advances in AI and machine learning, which have been used for structure prediction, they still don't work very well at predicting mutational effects, especially mutations which improve the stability of the protein. And that's because we don't have enough data for this. So we used, and this was before all the AIML stuff became popular, we used this methodology to identify stabilizing mutations in proteins, and there are a bunch of papers. So the data you get from an experiment is, if these are the residues in the protein, I told you proteins are between roughly 100 to 500 amino acids in length. This is a case where we have 100 different residues, and on the y-axis are the substitutions that we got at each residue. And the color code gives us a property which is related to the stability of the mutant. So in this single experiment, we can get data on, you have a 100 residue protein with 19 substitutions at each position, so 1900 substitutions. You don't necessarily get all of them, but you get at least 60 to 70%. The white squares are where we didn't have the mutant in the library. And so from a single experiment, you can identify a large number of potentially stabilizing mutations. Then we put these into the protein and express the individual proteins, and we characterize their thermal melting. And this is the thermal transition of the wild type, which happens a little bit above 60 degrees. And by adding additional mutants, we can considerably enhance the protein thermal stability. Okay. So now, this is the structure of the surface protein of the SARS-CoV-2 virus, called the spike protein. So all the vaccines that were developed, the thing that is the protective correlate is antibodies against this spike protein. Okay? The more antibodies you make that bind the spike protein, the better protected you are. In red is a part of the spike protein called the receptor binding domain, and the receptor binding domain binds to a protein called ACE2, okay, which is the receptor on human cells. So if we have antibodies that bind to the receptor binding protein and block the binding of spike to ACE2, then the virus will no longer be able to infect cells. So very early in the pandemic, we thought this would be a good candidate for a vaccine, and we showed that we could express this protein in a variety of different expression systems, and we showed very early on that it was highly thermal tolerant. So you could take this protein, which we expressed in mammalian cells or yeast, we could lyophilize it and take the lyophilized powder and expose it to boiling point temperatures, and when we cool the material down, it was still properly folded. But it wasn't, the wild type protein didn't produce large numbers of neutralizing antibodies when we injected it into mice. So we attempted to stabilize the protein through the methodology that I described, and this is the thermal transition of the wild type protein. This is of the mutant protein that we got. It increased by 8 or 9 degrees, and when we took this protein and put it into mice and then took the serum from the mice, then shown here as a log scale, the amount you can dilute the serum and still prevent virus from infecting cells. So with the stabilized protein, when we took the mouse sera, we could dilute it 100,000 times and the sera would still block the virus from infecting cells. But when we took the wild type protein, it was 100-fold worse. Okay? We took the same sera and then we looked at the Delta virus and later on at the Omicron virus, which came several years later. And these were all sera which were generated against against this stabilized wild type RBD. And you can see those sera still neutralize the Omicron virus, whereas the sera which were from the unmutated wild type sequence failed to mutate the virus. So just by stabilizing the protein by about 8 or 9 degrees, we could get a considerable enhancement in the ability of the immunogen, which is the RBD in this case, to produce antibodies that prevented viral infection. So we applied this methodology to many different proteins to show that we could stabilize several different proteins. And besides its use in vaccines, this kind of methodology is obviously good for when you want to make stabilized proteins for various bioprocesses, for enzymes, including enzymes used in laundry detergents, in various kinds of fermentation technologies, etc. But we have been focused on vaccines. So in red here is always the wild type unstabilized protein, the thermal transition, and in black is the stabilized protein for many different proteins. So it increases the stability. It also increases another important property, which is the thermal tolerance. So if you don't have access to good refrigeration, which is typically necessary for many vaccine formulations, then when the vial is exposed to temperatures which are different from room temperature, the protein loses its activity. So shown here is, this is from a different Sarbecovirus which caused an epidemic in the early 2000s. You can see that if it's stored at 4 degrees, it undergoes the thermal transition. But if we incubate the protein at 60 degrees for an hour and cool it back down, you can see the thermal transition has disappeared. Okay? And whereas when we take the protein that we stabilize through our methodology, you can see that it still retains the its stability even after keeping it at 60 degrees for an hour or two. And this was true for the surface RBDs from many Sarbecoviruses. And so the mutants show an increase in the melting temperature as well as protein yield, and later we showed that they also don't show a loss in the antigenicity. So SARS-CoV-2 is one of hundreds or more of different kinds of Sarbecoviruses. And these Sarbecoviruses are found in many different species. They are largely, most commonly found in bats. Okay? And so far there's been this epidemic in the early 2000s caused by SARS-CoV-1. There was the pandemic caused by SARS-CoV-2. So clearly they are capable of making the species barrier jump. And unfortunately, because of increasing population and continuous degradation of the environment, there is more and more contact now of humans with wildlife, including, many such species. And so many viruses which were formerly not coming in contact with humans do come. And in Kerala you have a classic example, which is the Nipah virus, right? So this is something, that's going to happen with increasing frequency. And these Sarbecoviruses are classified into many different clades. So there are at least 3 clades. The SARS-CoV-2 was from clade 1b, the SARS-CoV-1 was from clade 1a, and there are a bunch of other clades. And many of these bind the ACE2 receptor also. Okay? So we need, in addition to SARS-CoV-2, we need vaccine formulations that can protect against a broad range of Sarbecoviruses to protect against future pandemics, right? Because by the time a pandemic comes, it's too late to develop a vaccine, right? We saw, even though the Indian manufacturers did a fantastic job, by the time the vaccines really became available to the population, it was the middle of 2021, by which time the Delta wave had already started, right? So you need really vaccines to be, at least the designs to be prepared well in advance, and the manufacturing ability also to be developed well in advance. So we showed that for a bunch of different Sarbecoviruses, we could greatly increase the yield of the antigen by our methodology. So here the wild type was less than 50, it went up to 200, and the melting temperature also increased. And we showed that when we made a cocktail of these RBDs from different clades, it was quite immunogenic in mice, and it elicited neutralizing antibodies. What we next did was to fuse the RBD to the conserved subunit of the spike protein called the S2 subunit. Okay? And we showed that this particular protein expresses much higher yield, at a much higher yield than the whole spike protein. So we made these RS2 fusions with the RBD, and then we used them to immunize mice, and these mice are transgenic mice which express the human ACE2 receptor. So they are highly susceptible to infection by subecoviruses that bind to ACE2 like SARS-CoV-2 and the other subecoviruses. So what we did was to immunize mice, and then we challenged them with viruses that were not included in the receptor binding domains that we had immunized with, because we wanted to see if our vaccine would protect against challenge with a virus where the RBD was different from the virus we had immunized with. So we took the SARS-CoV-1 virus and the BA5 variant of the SARS-CoV-2. And when we did our immunizations and we looked, the unimmunized mice rapidly lost weight, and we had two different formulations, and these mice didn't lose weight at all. These are the control mice which were not challenged at all. And you can see that there was, with either challenge virus, there was very little weight loss, and this particular formulation, where we took the RBD fused to the conserved part of the spike called S2, showed the best result. These are the amount of virus that was left in the lung after challenge. So you can see in the unimmunized mice, you have of the order of 10 to the 7 copy number, whereas here it's gone down to less than 10 to the 2. So it's a 100,000-fold reduction in the virus titer. This is for SARS-CoV-1 challenge. This is for a heterologous SARS-CoV-2 challenge. Okay. Another thing we wanted to use this methodology for was to map epitopes in sera. Now, when a person is either immunized or infected with an infectious organism, as I mentioned, they produce antibodies, and these antibodies will bind to the surface protein of the infectious organism. And if we talk about viruses, there are typically only 1 or 2 important proteins on the surface. So it's important from a vaccine standpoint to be able to find out where on this molecule the antibodies are binding. It's a molecular recognition problem. So in most molecular recognition problems, you have 2 molecules which are binding to each other, and you want to find out the interface where the binding happens. But in this kind of situation, you have one antigen, but you have antibodies which are potentially binding at many different parts of the surface, because the immune response is what's called polyclonal, right? You get all kinds of antibodies. So it's important for us when we are using a vaccine and when we want to understand how protective it will be or what is the basis of protection, we must be able to figure out where on the surface of the virus these different antibodies are binding. And you can't do this very well by crystallography or cryo-electron microscopy or other techniques, because those are well suited to working with highly purified samples, where you have only 2 kinds of molecules, right, which are binding to each other. Here we have many different kinds. And so we used a methodology that we had developed, we and others, to address this issue. So what we do is we express the antigen on the surface of a cell like a yeast cell, and we make a mutant library of that antigen where we vary the identity of the amino acids on the surface of the antigen, and then we do through FACS or through other means, we find out which are the mutations which are preventing the antibodies from binding. And those mutations are ones must be at sites where the antibody was binding to the wild type protein. Okay. So we developed a methodology to do this in a simplified way. It had already been developed by others, but those libraries were much more complicated. So the idea of yeast display I've already talked about. So if there are residues which are in the epitope is the part of the antigen which is binding the antibody, those proteins will be expressed on the yeast surface, but they won't bind to the antibody at all, or to the sera in this case. So by collecting these yeast cells and sequencing the antigen that is present in these cells, which we can do very easily, we are able to identify all the regions on the protein surface where the antibodies in the sera have bound. Okay. So we did this, we got sera from, through our collaborators, we got sera from people who had been infected by the SARS-CoV-2 virus at the border of the original wild type strain and the Omicron strain. So that was in early 2022. And we used our methodology to find out where on the surface these are sera from 3 different individuals. And this is the amino acids on the spike protein. And these in red are the residues that we identified as being targeted by the antibodies. And the interesting thing was, in these 3 different people, the regions that were being targeted were pretty similar. And we could, because we knew the structure of this protein, we could map it. And what we showed was that these residues were in relatively conserved regions of the protein. They hadn't mutated very much. So what we predicted was that once we had identified these residues, that these sera would be able to protect against a virus that hadn't emerged at the time, that came out much later, almost a year later. And we confirmed that by doing studies, because we had those sera and we could look at whether they neutralize the so-called XBB virus, and you can see that they all neutralize the XBB virus, though with less titer than they neutralize the wild type virus. Now this is important because it's quite different from what others had seen with sera from people who had been immunized with mRNA vaccines in the US. Because for them, those sera didn't neutralize the XBB virus at all, or very, very poorly. So what's different in India, of course we got a different vaccine, but I don't think that's the main reason. The population that we sampled was A, much younger, because the average age in India is younger, and B, it was being continuously exposed to the virus. Because as you all know, apart from the very stringent lockdown that we had in the initial stage of the pandemic, you know, maybe in Kerala it was a bit different, but in most of the country after that, you know, there was very little enforcement of any kind of isolation. And so people were being continuously exposed to the virus. And because of this continuous exposure, I think the immune system response was broadened, so it started targeting less variable regions of the spike protein. And so these are the regions that we identified from the methodology that I talked about, and they are located in the face of the RBD that is different from the path that binds the ACE2. So most of the mutations, the early mutations happen in this region, around the ACE2 binding region. But the ones we got were quite far away. And the fact that these sera were able to neutralize the virus which came much later, perhaps gives us some insight into why there has been, even in the absence of vaccination, no significant uptake in the number of deaths at least. Infections are ongoing, and I still believe that it would be a good idea to have vaccines for those who need it, namely for the elderly and those with comorbidities, which we don't have in the country. Now, I'll briefly talk about, in the last few minutes, about influenza virus. So there are multiple clades of influenza virus. Right now, the clades that circulate in humans are H1, H3, and flu B. And there are influenza vaccines, but they don't work very well. So every year, a very large number of people die of influenza. And in pandemics, this number can be much larger. In 1918, there were almost 100 million people who died of flu in the pandemic, and 30% of them, or close to that number, were from India. So currently there's a lot of concern about an avian origin viral strain. You see, flu has multiple hosts, and there was an avian strain which has now crossed over into mammals. It's called H5N1, and it's caused over 500 million deaths in birds, and it's in more than 70 different mammalian species. So it's, you know, it's clearly a potential pandemic threat. And what we did was, several years ago, was to take a conserved region of the hemagglutinin surface protein, and by protein engineering we expressed this by itself, which had not been possible to do before that. And we showed that mice immunized with this conserved region were protected against viral challenge with quite a distant strain of the flu virus. So in 2017, I co-founded a startup called Minvax to take some of this work forward into the clinic. All our earlier work was done in collaboration with Merck, but then we decided we wanted to have the ability to do things independently, and so we express this part of the protein in insect cells, and we've developed a recombinant adjuvanted vaccine, which in all our laboratory tests is better than the inactivated virus-derived vaccine, which is what everybody gets and which is what is available in India also. So more than 95% of vaccines are made for flu from inactivated virus, which is grown in eggs. It's a very old technology. So this, and here we took this recombinant material, formulated it with an adjuvant. We've carried out a phase 1 clinical trial in Australia, and we have shown that this formulation is safe and well tolerated, and we are starting trials in India this year in collaboration with the Indian Council of Medical Research. So just to summarize what I talked about, hopefully to convince you that we are able to stabilize proteins relatively rapidly, and this enhances the immunogenicity. We got a lot of publicity for some of this work in the pandemic, but no funding, unfortunately, to take it forward in a timely way. There were issues with both prompt funding and also the kind of regulatory approvals that were required because of the regulatory systems that we have here. So we couldn't actually take this forward into the clinic, though I think it would have been good if we had been able to do that. So, but now we have taken this forward to develop vaccine formulations that we hope will protect against many different sarbecoviruses. We also have worked on and are working on other pathogens, including RSV and Nipah. And so we'll see how this goes. But if we are successful, then in probably 3 years' time, once all the trials are over, we should have a recombinant influenza vaccine, if everything works. Okay, so just a few overall remarks. So there have been a lot of success in AI/ML based approaches at predicting structures and doing many, many other things. Despite that, their predictions for stabilizing mutations have been moderately good, but they could be much better, and we are trying to see how we can improve those predictions. And although there's been a lot of enhancements in our ability to rapidly make vaccines, including the mRNA vaccines, there are still many things that we don't understand. And the rate limiting step many times is finding the right molecule to put into the vaccine. And other things, we have a very competent pharmaceutical industry that can take things forward. So we have very great strengths in biopharmaceutical manufacturing, and it was really demonstrated in during the pandemic when all our vaccines could be made within the country. But there are still a lot of significant regulatory and funding hurdles to move discovery research, especially from academia. It may be different in CSIR labs, but to move this into the clinic. But we have been able to do this, of course, only because Minvax was there. So I'd finally like to thank all the people in my lab over the years and some of our collaborators, Shashank Tripathi and Somnath Datta at IISc. These are Dr. Rajesh Ringe at CSR IMTech and now at NIPER for doing all the pseudoviral assays, collaborators who provided the adjuvant, the Bill and Melinda Gates, now the Gates Foundation for funding, Dr. Arindam Mondal at IIT Kharagpur who gave us the breakthrough infection sera with his collaborators, and NII which did the challenge studies with SARS-CoV-1 because we didn't have access to that. We got the transgenic mice from in CBS and in STEM, and Minvax, which has been really instrumental in moving a lot of this work forward. And this is the funding that we got. And for those of you interested, this was all the publicity that we got for our worm vaccine. But again, unfortunately, we couldn't really take it forward in a timely fashion. So I'll stop here now. I'm happy to take any questions that you might have.
Yeah, now it's open for questions. If anyone is having any queries, please let us know.
Sir, excellent talk. Besides inactivated vaccines, most of the vaccines that you have shown are macromolecules. So in a discovery point of view, where does the small molecules fit in vaccine development?
The small molecules are normally, the place they are most important for, of course, as additives to stabilize vaccines. Also for the mRNA vaccines, all the lipids are, very important components of mRNA vaccines, and also in adjuvants. So they are small, equivalent of small molecule sized molecules that are used as adjuvants to stimulate the immune system. So those are the main areas where small molecules are useful in the vaccine context.
These adjuvants, can you give some examples? Which are the adjuvants that you have used?
So, okay, so now because we are really focused on human use, right? And you may be surprised to know that there are very, very few adjuvants that are licensed for human use. The majority of vaccines use alum, okay, or different forms of alum. There is also, and what we have used is an oil in water emulsion, which was originally developed by Novartis, called MF59. So the problem with non-alum adjuvants is they have all been guarded by pharmaceutical companies. They are not available in the public domain. So the Gates Foundation funded the Vaccine Formulation Institute to make equivalent versions of some of the approved adjuvants. So the adjuvant that we use, called SWE, is equivalent of MF59, which has been used in tens of millions of people. And then there are other adjuvants, but they are many of them are proprietary, like AS01, AS03, and some of them have, as one of the components is, they are saponins, which are important components. MF59 has squalene, which is from sharks. And then there are dinucleotides like CpG, which have also been used as adjuvants. So there are, but again, the ones that have been in humans are probably not more than 5 or 6. And of them, it's really only alum and now MF59 that are available in GMP grade for other people to use. And maybe a couple of others will soon be available in GMP grade.
Thank you.
Thank you, sir. Thank you for the wonderful talk. My question is related to the thermal tolerance or the melting temperature enhancement of these proteins. Is it because of the compactness induced by the conformational change, or the interactions will play a role?
So the reasons for the enhanced stability are the mutations that we have made have introduced new molecular interactions into the protein, which makes it more stable.
There is no conformational change basically. There is no conformational
There's no
change.
conformational, yeah, because so the thing is that you want to put very few mutations because you don't want to change the surface of the protein significantly, and you want the shape of the protein to be retained. And we check all that because we have antibodies which are conformation specific.
Okay, thank you.
Yeah.
Sir, very nice talk, very informative. So my question is, when you are infected with this modified temperature sensitive protein to the mice, so what was the changes in the humoral immunity and the innate immunity? Like, is there any changes when it is administered to the mice?
Well, there are two things that we haven't looked at the cellular immunity very much, and we don't expect it to change also too much. The humoral immunity was different in two ways. The titers of antibodies went up slightly, but more important, because we used our methodology to map which parts of the surface were targeted by the antibodies. So it appeared to be targeted to more conserved regions of the protein. So we don't fully understand why stabilizing the protein improves the antibody response. One of the reasons, probably, there are two reasons which are, I think, conceptually simple to understand. The first is that the proteins, although you see them as a static structure, actually they are undergoing a lot of dynamic fluctuations. And these dynamic fluctuations expose parts of the protein to the immune system that are not the native conformation. And then you get antibodies against those conformations. This is well established for HIV, for the HIV envelope protein. So if you stabilize the protein, make it more rigid, then probably you get less of these fluctuations. The second possible reason is that once you inject the protein into the body, it gets degraded over time. And maybe having a more stable protein makes it last a little bit longer in the body, though we don't have direct evidence for that.
Okay, thank you, sir.
Sir, I have a question. Sorry, can I?
Yeah.
Sir, thanks for the wonderful talk. So from your talk, I understand that fundamental to the immunogenicity of a protein or the antigen is the structural regions. And this is also where the mutations are happening when the virus is evolving. So shouldn't it be theoretically possible that we design vaccines anticipating these mutations so that we can have the same vaccine which is which having different sets of epitopes and have an anticipatory immunogenicity profile?
Right.
Yeah.
So the problem is that viruses can mutate in all kinds of ways, and so it's very difficult to predict in advance which you might even know the regions of the virus which are mutating, but to predict exactly what mutation is going to happen is virtually impossible. So, and unfortunately, even one or two amino acid changes can significantly affect the ability of the antibodies that were generated to neutralize the virus. So if you look at the Delta virus, the number of mutations on the spike is relatively small. It's only like 3 mutations or something. But still, the antibodies that were raised against the wild type virus were significantly less against the effective against the Delta virus. And then when it came to Omicron, one of the big things that before that, all the mutations had been single point mutations. In Omicron, there were simultaneously about 10 or 15 mutations. And we still don't understand what was the driving force for that. Whether the virus first went into another host and then came back, or whether it infected an immunocompromised individual who couldn't clear the virus and then it just mutated. So it's not been been feasible to do that. That's certainly a good suggestion. The other approach that people have been taking is to try and somehow target the conserved regions of the surface. So in the case of flu, for example, that is what we did, right? So we made an immunogen that targets, that is only this part. We cut this out of the rest of the protein, and we showed that we could still, through protein engineering, get it to fold up. And we got antibodies that were protective. Now, the thing is, why is this protective? Why is this conserved? Why isn't it mutating? There are two reasons. One is this is important in some functional aspects, but so is this is the part which binds the receptor. So there are conserved parts here also. But the other region it's conserved is that on the flu virus, these hemagglutinin proteins are very closely packed together. And so this region is not very accessible to the immune system. So while we get antibodies that are able to protect, their potency is not very high. So there are lots of strategies that the virus uses and reasons why some parts are protected and less accessible to the immune system that make it difficult to even target conserved regions of the surface protein. But it's a very active area of research, and people are trying to figure this out.
Thank you, sir. Thank you very much for such a wonderful and informative talk. My question was something related to what Dr. Rajiv he asked just now, because we know that the virus undergoes mutations very fast, which is something which we have already experienced during pandemic. So how difficult or how easy to come up with a alternative vaccine to take care of this kind of mutations? That is my first question. Second question is Nipah virus. It is known for long time, and what is the bottleneck in coming up with a vaccine for Nipah?
So there is zero difficulty in coming up with a vaccine for Nipah. Okay, we had a small amount of funding. A student in my lab worked on it for a little bit, and we have things that at least are highly immunogenic. And we produce antibodies that neutralize the virus in cell culture. The problem now becomes who is going to take this forward, okay? Because now once you have a candidate antigen, first we have to do a challenge. Let's say we overcome that. But then there has to be a company which is ready to manufacture the vaccine. And why will a company manufacture a vaccine for an infectious disease that comes, once in a while? It infects, a very small number of people, and they die and they don't transmit it, right? So the only way for such vaccines to be produced is the government has to make it a strong focus, and they have to give a contract, right? You can't expect a company to produce this as a commercial product, because it's not going to be a profit-making product. There has to be a separate, development pathway by which the government decides that this is important to do, and it just keeps this available. But again, so here, for example, if there were a Nipah vaccine, how many people would take it? Nobody, right? And you're living in a state where Nipah is happening every few years. Now, if none of you are willing to take it, who's going to take it, right? Because the vaccine has to be given in advance of the infection. There's no point where, so that, you have your answer.
Sir, I have a quick question related to that Robbie asked. As you mentioned few of the adjuvants, one related to yours is alum. So
ours is not alum, ours is the MF59. Yeah.
So what about, sir, there are many antigenic oligosaccharides are there,
right?
Which is also established in many cases. So that could be one of the adjuvants in in a common way, not only influenza, but what is your
Sure.
comments?
So the problem again is not, so you know, people have made hundreds of different potential adjuvants, okay? But now the question is, who, see, if you want to put this into people, they have to be GMP manufactured, okay? Then you have to do a safety trial and to show that the vaccines are safe. In the pandemic, people were dumping all kinds of things into you know, into people, that so, and the regulators were didn't object. But now at this time, right, let's say you make a potential adjuvant, the GMP manufacture costs some amount, but even if you have that, then you have to run a clinical trial. And you know, again, those clinical trials are expensive, and the difficulty is that in an adjuvant, you need to balance the ability to induce an immune response versus its ability to induce too much of a response, because that also can be damaging. And so, you know, who's going to take the risk? Because these things cost tens or millions or much more than that. So somebody has to take the risk and the liability, and you know, it's not clear who's going to do it. That's why there's so few adjuvants.
Thank you, sir. I'm sure there are a lot more questions, but due to lack of time, we have to end the question and answer session now. So we thank you, sir, for sharing your glimpses on your research on vaccine development and also encouraging the young minds. CSIR NIST hosts more than 250 PhD students enrolled with ACSIR who are working on several cutting-edge research on wide array of research domains. Representing all these PhD scholars, we have a science club in the institute consisting of one member from each department. The science club has been revised recently due to completion of PhD program of the former members. As part of the National Science Day celebrations, the ACSIR Science Club conducted 2 competitions for the whole students community of NIST. Now we request the president of ACSIR Science Club, Mr. Harikrishnan PC, to announce the prizes for the competition.
Good afternoon to everyone present here. A warm welcome to all the dignitaries, faculty members, research scholars, and students gathered to celebrate National Science Day 2026. As part of the celebrations, 2 major competitions were conducted: Science Quiz competition, AA poster competition. Coming up next is the award ceremony. I request our Honorable Director Dr. C. Anantharamakrishnan and our Chief Guest Professor Raghavan Varadarajan to do the honors. Please welcome. Thank you, sir. The science quiz was conducted on 26 February and it was an exciting and intellectually stimulating event. We extend our sincere appreciation to the quizmaster Dr. Juby John for conducting the quiz with a great energy and precision. We will now announce the winners. I request Professor Raghavan Varadarajan to kindly present the awards. The 3rd prize goes to Team C set. I welcome the team members Aditya C. Anand, Sangeeta Skinny, Suryasri SR to receive the cash prize. The 2nd prize goes to Team ETD. I welcome the team members Krishna JS, Ram Keshav and Tamjit Naushal. The 1st prize goes to Team BBD. I welcome the team members Amber Sahi, Geetanjali Sahu and Ajay Pradeep to receive the cash prize. Our second major event was the AA poster competition conducted under the theme My Lab Visualizing Research through AA Prompt. We received an enthusiastic response with a total of 20 posters. The posters were evaluated by an expert panel, Dr. K.V. Ramesh, Dr. V. Arun Kumar, and Dr. Akshay Dilip Shinde. Before announcing the names of winners, I request our director, Dr. C. Anantharamakrishnan, to kindly present the awards. We have a consolation prize. I welcome Mr. Sangeeta Skinny from CSET. The third prize goes to Ms. Maria KS from AFPTD. Second prize goes to Ms. Adhina M from AFPTD. The first prize goes to Ms. Trisha Fernando R from AA&ML. Congratulations to all the winners of both the science quiz and AA poster competition. We also extend our appreciation to every participant for showcasing their creativity, scientific spirit and enthusiasm. Thank you.
Thank you, Hari Krishnan. It's now time to honor our chief guest by presenting memento. I request our director to kindly present our chief guest, Professor Raghavan Varadarajan, with a shawl and a memento. Thank you, sir. It's time to convey our heartfelt appreciation to everyone who helped in organizing this National Science Day celebrations at our institute. May I now request Dr. Joshi Joseph, Senior Principal Scientist, CSIR NIST, to propose vote of thanks.
Respected Director Dr. C. Anantharamakrishnan, distinguished Chief Guest Professor Raghavan Varadarajan sir, esteemed dignitaries on and off the dais, colleagues, students and dear participants, a very good afternoon to all. It's my pleasant privilege to propose the vote of thanks on this special occasion of the National Science Day celebration at CSIR NIST. First and foremost, we express our sincere gratitude to our Chief Guest, Professor Raghavan Varadarajan, for delivering an inspiring National Science Day lecture. Sir, thank you very much for taking us through the journey of vaccine development in your lab, and thank you for graciously accepting our invitation and enriching this celebration with your presence. We convey our heartfelt thanks to our director Dr. C. Anantharamakrishnan for his visionary leadership and for continuously fostering a vibrant scientific culture at CSIR NIST. Sir, your encouragement enables us to celebrate science not just as a profession but as a societal mission. Thank you, sir. Our sincere appreciation goes to Dr. TPD Rajan, Head Human Resources and Academic Division, for the warm welcome and for the constant support extended by the HRID team in organizing these events smoothly. celebrations like National Science Day are always collective efforts, and we gratefully acknowledge the contributions of several divisions and teams whose dedication worked behind the scenes. The HRID team members for meticulous planning and coordination, the HLS and ITSU team for their technical and logistical support, the administration and IMAC members for ensuring smooth arrangements and institutional coordination, the canteen and guest house management and staff for their hospitality and arrangements in organizing, welcoming and making arrangements for the participants and guests, the ACSIR Science Club and its enthusiastic office bearers for their active involvement in organizing and engaging students, the Jigyasa team for promoting scientific outreach and strengthening interactions with young minds, and all divisions for making arrangements for the open day visit today. Our sincere appreciation also goes to the student volunteers whose energy and commitments made the open day visit a great success. Our campus is hosting around 500 students from various schools and colleges as part of the open day today. We extend our special thanks to the students and faculty members from various institutions who visited us today. Your curiosity, enthusiasm, and active participation truly brought life to this celebration and strengthened our shared commitment to nurturing scientific temper among young minds. We also thank all the faculty members, researchers, staff, students, and all the members of CSIR NIST who participated wholeheartedly and contributed to making today's program meaningful and vibrant. Finally, we extend our heartfelt gratitude to everyone present here, in person and online, for joining us in celebrating science and reaffirming our commitment to scientific temper and innovation for societal benefit. With these words, I once again thank each one of you for your support and participation. Thank you.
Thank you, Dr. Joshi. As we conclude, I request everyone to rise for national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे